Hello, everybody. My name is Zhichao Xia, and I work at the University of Connecticut. My research interest is using neuroimaging tools, especially multimodal MRI, to understand individual differences in language and reading skills. I have included my contact information on the screen. Do not hesitate to reach out if you have any questions about neurolinguistic or MI data analysis. Today, we are going to talk about data structures. Before that, I'd like to give you a general idea of where this question comes from. As cognitive neuroscientists, most of our work focuses on data. Of course, since we are interested in different research questions and plan to address these questions in specific ways, the data we will use will be very different as well. For example, if we ask whether the left superior temporal gyrus is involved in word reading, the data we will use will differ from what we will use to answer the question whether phonological processing skill in the pre-literature stage contribute to reading development during school years. Here, we pause for 10 seconds so you can quickly think about this. Okay, just pause the video if you need more time. So, to answer the research question, the first idea I believe that will come to my mind is to design a new experiment, collect data with fMRI, perform data analysis, interpret the results, and write up papers to share the findings. We can also search for published papers related to the topic, extract information from the research reports, and conduct a systematic review or meta-analysis. Besides, given that some researchers, for example, senior members in our lab, may have already done relevant research before, we can request access to the existing data sets. However, in most cases, and in my cases, when an undergraduate student joins a lab or a graduate student just starts their PhD journey, they can hardly start a new neuroimaging experiment immediately. We usually start by conducting a literature review, meta-analysis, or mega-analysis if possible. I have done some, and my experience is that extracting information from the paper can very time-consuming, especially in the past when there was a lack of guidelines about reporting the results. Under such circumstances, accessing the related data stored in a specific framework will significantly reduce the workload. One important aspect is that we will make fewer errors since they are clearly stated, and there is no need to guess. After the review, meta-analysis, and mega-analysis, we will have the first answer to our research question. More specific hypotheses may also be there. Thus, we move to our second approach, using available datasets. It can come from either existing datasets in the lab or publicly available datasets. The story is when I first accessed the actual fMRI data in the lab around 2010, I was very excited, but soon became worried as I realized that there was more to do to understand the datasets, including but not limited to figuring out, first, the background, original research questions and hypotheses, second, details about the research design, tasks, and stimuli, third, parameters of the neuroimaging data collection, fourth, expected and unexpected events during data collection, and fifth, missing data and any amendments as the study progressed. To resolve this, I asked the senior lab members in charge of the project for help. We had several meetings, information exchanges, and email confirmations. I did learn a lot. Here is an example about the information that needs to be confirmed. In analyzing fMRI data, one important aspect is the sequence parameters. Usually, the parameters are stored in the heads of the raw DICOM files. Okay, so here is a screenshot of a small part of the parameters using the MATLAB function DICOM info. What I did then was first double check the parameters. Second, write them down, and third, feed them to the pre-processing steps manually. At that time, I thought it would be better to organize the data in the same way and add as much information as possible to describe it. In addition, it would be better if the software or pipeline for data pre-processing could recognize the parameters automatically. What we did at that time was create a document containing the steps, such as how to convert MI data, how to name the data, how to put information about the study and data collection in a text file in the folder we created for the project. We followed the document each time new data was collected. Later on, when I heard about BIDS, I realized that it is actually an issue faced by all the researchers working with neuroimaging data, and it is great that the Stanford team developed such a useful framework to handle the data. We may face the same situation when accessing data from other labs or platforms. On the one hand, it is wonderful that the authors are willing to share their data. On the other hand, there could be much more work if the data is stored in a disorganized manner and contains missing information, which means it could take several actual months to understand what the data is. And if there are any issues, attention should be paid to them. Thus, the same point remains true. If the data is stored in a well-established way that is used by the community, it will save a lot of time and significantly promote progress. With the findings from the meta-analysis and mega-analysis and the work with the public datasets, we now have a better idea about the experiment we will design to answer our research question. As we are in charge of the study, administration, data collection, and curation, we can certainly decide what we will do with the data as long as it has been approved. Even so, a standard framework will also be helpful. Here are some reasons. First, it can help us gain a comprehensive understanding of the data, the study, and what we will do next, for example, in data analysis. Second, we may need to revisit the data months or years later. It can help us retain important information. Third, as mentioned in the second point, lab members may want to use the dataset to answer the questions they are interested in. Thus, it will be easier for them to understand the data if there is a standard structure. Fourth, given that new trends and sometimes actual requirements by the grant or institute, the dataset needs to be started with the community, which makes the standard data management approach very important. And fifth, even though it is not straightforward, it can also benefit systematic review, meta-analysis, and mega-analysis. All right, from these examples, we know that a standard data structure and curation strategy can always benefit any research approach. So today, besides trying different strategies in our own way, the brain imaging data structure, or BIDS, can help. Since we have discussed several examples, let's move to more general statements. First, the motivations. Data sharing has become popular not only in our own labs or universities, but also worldwide. This commonly happens, first, between research institutes, different researchers at the same institute or even within the same lab. Researchers sometimes use the same dataset to compare toolboxes and pipelines and test novel approaches. The reason why data sharing is so popular and important, including first, more information or knowledge can be gained from the data. Second, the producibility of the findings can be examined. Third, it can create a scientific playing field, allowing students and researchers who don't have the machines to conduct neuroimaging studies as well. Fourth, we can have larger and more diverse training datasets for advanced data analysis, such as machine learning. The increased need for and the practice of data sharing also calls for increased efforts to create a high demand for neuroimaging data management and curation, especially given that multiple techniques and modalities have been and will continue to be included. However, the differences in data organization, and data description causes multiple issues when we share data, and extra meetings are needed to address these challenges. Moreover, even after figuring out the answer, it will take significant time to validate the data. The issue can be addressed at different levels. For example, within the lab, team members can follow the same in-horse guide to handle the data and input the necessary information. The same strategy can also be adopted when cooperating with other labs and the entire community. Pioneer researchers took actions when they launched their famous projects, such as FMI Data Center, International Neuroimaging Data Sharing Initiative, Functional Connectome Project, Human Connectome Project, and most recently, to my knowledge, the largest neuroimaging dataset in the US, ABCD, Adolescent Brain Cognitive Development Study. In this project, researchers tried to use the same standards across sites where the neuroimaging data were collected. Through these efforts, researchers learned several principles to establish a framework that will promote data sharing, referred to as FAIR. Findable data means sufficient metadata. There should be unique and persistent identifiers, and it must be registered for indexing in a searchable resource. To be accessible, metadata and data should be readable by humans and machines, and they must reside in a trusted repository. In terms of interoperability, data must share a common structure, and the metadata must use recognized formal terminologies for description. Finally, data collection must have clear usage lessons, clear provenance, and meet relevant community standards for the domain. We can clearly see a standard data structure is necessary here. As one key element in the data structure, standard file format that allow researchers to reuse data across multiple software and pipelines have been developed. If you worked with neuroimaging data in the old days, you may know that there are three major software for analyzing fMRI data: AFNI, SPM, and FSL. And you may also know that they have their preferred data formats. For example, AFNI uses pairs of files named head and brick. SPM uses Analyze 7.5, which also consists of a pair of files, .hdr and .img. FSL prefers .nmi.gz. Particularly, the head and brick files that AFNI uses are specific to AFNI itself and cannot be recognized by other software. Also, the old versions of AFNI could not directly handle other data formats either. We need to convert the data format during which errors could occur. These issues were later addressed as the newly released AFNI accepted the nifty format. Data format is not the only thing we need to make standard. The other types of data and associated metadata need to be organized in a standard way too, so that data recipients can directly and quickly and accurately understand the details of the data. With this standard, first, the burden of curation can be minimized. Second, the likelihood of errors can be reduced. Third, we can develop tools that can directly utilize the data structure and validate the data. Against this background, one of the most featured structure, Bids, was born. On the screen is a Bids history timeline, several general ideas we can gather. So first, Bids is very young, less than 10 years since the idea was introduced in 2015. Second, in the beginning, This was more about MRI, especially task FMI. Then it was extended to handle more modalities, including but not limited to MEG, EEG, and FNRS. Third, the project is still ongoing, given the fact that more neuroimaging modalities have been developed and more multimodal researchers has been conducted to better understand the brain and its functions and structures features. The idea to develop a standard data structure came from Dr. Russell Podrack's post on October 17, 2014. Since then, Dr. Podrack, Dr. Grogolowski, and Dr. Bach have started working on the project supported by the Laura and John Ammon Foundation at Stanford University. This work is part of the effort to move from OpenFMI to OpenNeural. In the new OpenNeural platform, all datasets uploaded should be in the bits format. In the OpenFMI project, the team faces the same data curation issues as you and I did, but in a much larger scale. To deal with it, the data was reorganized in a OpenFMI.org style. Importantly, such data types can be directly imported to FSL and PyMVPA. The point is very crucial because users, for example, cognitive neuroscientists, will almost always choose the option that benefits their work with as little effort as possible. If it's just a certain format that is not directly supported by any software, it may turn out that nobody will use it. However, the personnel spent significant time ensuring and the validation of the uploaded dataset and transforming it into a certain and consistent form. The rapidly increasing number of datasets may make the labor inbound increasing steeply. What's more, these studies using specific modalities and data analysis pipelines, making data curation and validation almost impossible. In this case, a straightforward thought could be to encourage the author to ensure the data format follows the requirements of the platforms. As the amount of data in a single study is very small or is small, the authors of a certain project may only need to spend a short amount of time. Based on the experience gained from the Neuroimaging Data Sharing Task Force under INCF and the Neuroimaging Data Model Project, BIDS was formally introduced at a January 2015 meeting at Stanford. The photo shows the original idea that was written on the whiteboard. Starting from that concept, the team drafted the first version of the BIDS specification, prepared several example datasets, and developed an early version of the JavaScript-based BIDS validator. With BIDS, on the one hand, the dataset owner can transfer their dataset according to the guidelines and examples, with the platform where the platform admin can validate the uploaded dataset using the tool. So, what is BIDS? It stands for Brain Imaging Data Structure, which provides a standardized format for organizing and describing neuroimaging data. It consists of three essential parts. First is a nested directory structure. Second, file name conventions. And third, a JavaScript object notion sidecar, which is also called JSON. On the screen, I put a link to the official web page. 1 year later, the first official version was released. Let's first see what it does. Okay, on the left side of the screen, we can see raw DICOM data. That's exactly what you will get from the scanner after you run the experiment someday and download them in the evening. The right side of the screen shows that the data has been sorted and organized in bits format. As you can see, there is a list of participants. Under the subfolder for each participant, There are folders named for specific modality. In each subfolder, we have the brain imaging, and we have brain images in compressed nifty format, that is .ini.gz, along with the sidecar, that is the JSON file, where the parameters of the sequence are stored. After the general view, there are several key implementations. So the first, some metadata is encoded in the folder structure. Second, some metadata is replicated in the file name or for simplicity. Third, use tab-separated files for tabular data. Fourth, use compressed nifty files for imaging data. Fifth, use JSON files for dictionary tab metadata. Sixth, use legacy text file formats for B vectors and values, and that is the diffusion data and physiological data. 7. Make certain folder hierarchy levels optional for simplicity. 8. Allow arbitrary file not covered by the spec to be included in any way that the researchers deem appropriate. So there are several features of Bids are listed on the screen. First, Bids can handle multiple sessions and runs, which is always the case for neuroimaging studies these days. Second, it supports separate acquisition, sparse acquisition, are common techniques used in the auditory research to avoid influence from scanner noise. Third, it supports continuous acquisition covariates. Fourth, it supports multiple field map formats. Fifth, it supports multiple types of anatomical scans, for example, T1 and T2. Sixth, it supports functional MRI, both task-based and resting state. 7. It supports diffusion data. 8. It supports behavioral variables at the level of subjects, sessions, and runs. As mentioned before, Bids has been undergoing updates since the first day it was released. There are several key principles behind these updates. Principle 1. Adoption is crucial. That is, engage the relevant research community and keep the standard as close as possible to the practices already in use in the community. Principle 2. Do not reinvent the wheel. That is, use existing standards and file formats whenever possible. For example, Nifty, JSON, TSV. Principle 3. Some metadata is better than no metadata, which is very straightforward. Principle 4. Do not rely on external software or complicated file formats. Principle 5. 80-20 rule. There will be always edge cases that cannot be well accommodated by the standard, but it is fine as long as the standard works well for most people in most circumstances most of the time. All right, let's talk more about the updates. First, the field of the neuroimaging is developing rapidly. Thus, new modalities and sequences are always developed and included in understanding brain structure and function. To address this situation, base extensions have been developed to handle, for example, QMI, MEG, etc. Users can also contribute to this process via the base extension proposals. Second, base was originally developed to handle raw data. However, in terms of data curation and sharing, more and more researchers have started sharing preprocessed data or derivatives. This makes a lot of sense because for larger data sets such as ABCD, it is almost impossible for a single group to do all the data preprocessing, which takes a huge amount of time. Thus, sharing derivatives is reasonable. Another important reason is that a large group of experts will help develop the preprocessing pipelines. This can also make the findings from these derivatives more comparable. There are two major projects. First, the Bayes computational model framework, and second, the Bayes states model, which is a common statistical model that is applied to neuroimaging data. As for the present state, it is hard to know exactly how many researchers are currently using BIDs in their study, but it can be reflected in the increasing number of open neural data sets and the databases that have administered MIQC. As the figure shows, quite a lot of researchers have tried to use BIDs in their projects. Second, the growth rates are increased in recent years. Therefore, this is a highly successful example of community-driven standard for data organization. It is also one of the only grassroots standards to have gained such broad acceptance within this field. We talked before about the software that can directly handle a certain data format. In terms of the bits, these apps have been developed. Here is a table listing some applications. You can see that well-known software and pipelines such as FreeSurfer, HTTP pipelines, FMI prep, etc. Here is their website. Just go there for more detailed information. It is very helpful for researchers as you can use almost all the major software and pipelines with bits. Besides these applications, there are also online platforms that can run our data analysis with these bits apps. Two popular sites are Brain Life and Enigma. As for the challenges, the first one is delayed adoption by large data banks like the UK Biobank. However, this can be accepted because it is very hard to modify the data as there are tons of thousands of data points. Second, the conversion of bits used to be a headache. However, it can be easier now that several toolboxes have been released. There are specific toolboxes such as Bitcoins and certain modules in a toolbox, for example, Deparby. Third, there is a lack of machine-readable standards. Fourth, challenges of the BEP, that is, Base Extension Proposals Management exist. And the last one is that geographical diversity and inclusivity. Just look at the map. BIDS is most used in Europe and in the US. More work needs to be done to spread it around the world. Okay, summary. So first, BIDS is a standard way to organize data in cognitive neuroscience and related research fields. Second, most data types are well documented and the transformation can be quite easy and straightforward. Third, it can help promote data curation and sharing, whether within the lab or across multiple sites. Thus, just try it out in your study. All right, that's all the content for today. Next time, we may try some data transformation with different toolboxes, such as Bitcoins, as mentioned before. To prepare for this lecture, I've referred to multiple papers and presentations. I'll put them on the screen and would like to express my gratitude. All right, if you have any questions, just email me at zhichao.xia@ucon.edu. Thank you for listening. You all have a great one. Take care.